Welcome to Bioclass HUB, diving into C3 and C4 plants. Tighten your belt.
The fields are giving plenty this year. We harvest the maize, sorghum, and sweet sugarcane.
Photosynthesis in C3 plants. It consists of three main stages. 1. Carbon fixation. Carbon dioxide combines with a 5-carbon sugar called ribulose bisphosphate. The enzyme is ribulose bisphosphate carboxylase, which catalyzes this reaction, forming an unstable 6-carbon compound that immediately splits into 2 molecules of 3-phosphoglycerate, 3-PGA, also known as phosphoglyceric acid or glycerate 3-phosphate. Second stage, reduction. 3-PGA is reduced to glyceraldehyde, 3-phosphate G3P, using ATP and NADPH. Third stage, regeneration of RUBP. Some G3P molecules are used to regenerate RuBP using ATP, ensuring the cycle continues. Fourth stage, carbohydrate formation. A fraction of G3P molecules is converted into glucose, starch, or other carbohydrates. Energy requirement for C3 plants. To synthesize 1 molecule of glucose, the Calvin cycle must fix 6 carbon dioxide molecules. For every 3 carbon dioxide molecules, it yields 1 net G3P, a 3-carbon product. The cycle uses 9 ATP plus 6 NADPH. Therefore, to make 1 glucose, 2 G3P molecules are used. Therefore, 6 carbon dioxide molecules yield 1 glucose molecule. It's important to understand this. We shall find out that C4 plants use more energy than C3 plants. The low energy requirement gives C3 a competitive advantage under some conditions. In the image, there are examples of C3 plants: beans, rice, wheat, barley, Irish potatoes, groundnuts, soybeans, tomatoes, sunflowers, coffee. A common plant is the Irish potato and beans, grown in the Kabbalah regions of Uganda.
Look at this one, sister.
It is perfect. Our hard work pays off.
This plant has a competitive advantage in these cold and low-temperature regions of Uganda. Formation of lipids, fats, and oils. Glyceraldehyde 3-phosphate, G3P. G3P is converted into pyruvate and then into acetyl-CoA, a 2-carbon compound. Acetyl-CoA serves as the basic building block for fatty acid synthesis. Fatty acids combine with glycerol, which is derived from another G3P molecule, to form triglycerides or lipids. Thus, the carbon source for lipids comes from G3P from the C3 pathway, while the energy, ATP, and reducing power, NADPH, also originate from the light reactions. Amino acid synthesis for the synthesis of proteins. G3P and other intermediates enter glycolysis and the Krebs cycle, forming organic acids like pyruvate, oxaloacetate, and alpha-ketoglutarate. These serve as carbon backbones for amino acids. Nitrogen assimilation. Plants absorb nitrates or ammonium ions from the soil. Through the nitrogen reduction and assimilation processes, nitrogen is incorporated into amino groups. Protein formation. The amino acids produced are linked together by ribosomes during protein synthesis, translation, to form structural and enzymatic proteins. Pause the video and summarize this information. Now, let us dive into C4 plants. C4 plants, maize, sugarcane, and sorghum, have a special leaf anatomy called Kranz anatomy, which helps them photosynthesize efficiently, especially under hot, dry, and high light conditions. The leaf has 2 types of photosynthetic cells, mesophyll cells, arranged around the bundle sheath. Bundle sheath cells form a tightly packed layer around the vascular bundles. These 2 cell types cooperate in a process known as the C4 pathway, Hatch-Slack pathway. Dark reactions in C4 plants. The Hatch-Slack pathway in the mesophyll cells. Start with carbon dioxide fixation in mesophyll cells. Carbon dioxide first enters the mesophyll cells. It combines with phosphoenol pyruvate, a 3-carbon compound, to form oxaloacetic acid, a 4-carbon compound. This reaction is catalyzed by the enzyme phosphoenol pyruvate carboxylase, which has a high affinity for carbon dioxide and does not react with oxygen. The oxaloacetic acid is then converted to malate. Malate is shunted into the bundle sheath cell along the plasmodesmata, where it is then decarboxylated to release carbon dioxide, used in the Calvin cycle to form glucose. The pyruvate formed is then used to regenerate phosphoenol pyruvate with the use of energy, catalyzed by the enzyme pyruvate phosphate decanase. With this kind of pathway, more energy is used for the 2 cycles. 30 molecules of ATP are used. It has 12 additional ATP molecules are used for regenerating phosphoenol pyruvate. Examples of C4 plants include maize, sorghum, millet, sugarcane, amaranthus, and finger millet. These plants grow well in certain regions. Let us dive into the conditions that can affect C3 and C4 plants. Under normal circumstances, ribulose bisphosphate, carboxylase, fixes carbon dioxide onto the RUBP to form glyceraldehyde 3-phosphate. Photorespiration is the light-dependent process in which ribulose bisphosphate carboxylase oxygenates, leading to the release of carbon dioxide without producing sugars. This is a wasteful process. In conditions where oxygen concentrations or partial pressures exceed those of carbon dioxide, Rubisco burns the RUBP. It's common in C3 plants. Conditions favoring photorespiration. Low carbon dioxide level with high oxygen concentration. Rubisco binds to oxygen instead of carbon dioxide. High temperatures. High temperatures increase the likelihood of photorespiration. C3 plants, e.g., wheat, potato, soybean, rely on Rubisco for carbon dioxide fixation. In hot, dry, or high light conditions, C3 plants are more prone to photorespiration, which reduces their photosynthetic efficiency. That's why C3 plants can't grow well in lowlands with high heat, dry conditions, and high levels of oxygen. This is a list of C4 crop plants in Uganda and the areas where their productivity is high. Significance of the arrangement in C4 plants. Spatial separation of carbon dioxide fixation and Calvin cycle. Fixation occurs twice. The light reactions that produce oxygen do not occur in the bundle sheath cells. This reduces photorespiration. High carbon dioxide affinity, no photorespiration. The enzyme PEP carboxylase has a high affinity for carbon dioxide to concentrate it in the bundle sheath cells. High carbon dioxide in the bundle sheath cells enhances Rubisco efficiency to fix carbon dioxide without photorespiration. Reduced photorespiration, less energy and carbon wasted. Water use efficiency partially closes the stomata to reduce water loss to increase productivity during drought conditions. Advantages of C4, photosynthesis. C4 plants are highly adapted to hot, dry, and high light environments due to specialized features that enhance carbon fixation and water use. High photosynthetic efficiency. PEP carboxylase has a strong affinity for carbon dioxide and is not inhibited by oxygen, enabling rapid and continuous fixation even at low carbon dioxide concentrations. Reduced photorespiration. Cran's anatomy and the malate shunt concentrate carbon dioxide in bundle sheath cells, minimizing oxygen interference with Rubisco water conservation. Stomata can remain partially closed, reducing transpiration losses. C4 plants lose about half the water per carbon dioxide fixed compared to C3 plants. Heat tolerance. Adapted to high temperatures and intense light, C4 plants maintain photosynthesis, where C3 plants become inefficient. Structural adaptation. Bundle sheath chloroplasts, with reduced grana, lower oxygen production, and further suppress photorespiration. Effect of altitude on the abundance of C3 and C4 plants. C3 plants. In high-altitude regions of Uganda, such as Kabale, Kisoro, and Kapchorwa, temperatures are low and oxygen concentration is slightly reduced. These cool conditions reduce photorespiration, allowing C3 plants to photosynthesize efficiently. Enzymes like Rubisco perform best at such moderate temperatures. Result: C3 plants are more efficient in the highland regions of Uganda. C4 plants. In these same cool highlands, C4 plants still fix carbon dioxide effectively, but gain less advantage since photorespiration is already low. The extra ATP energy required for the C4 pathway makes them slightly less efficient in these conditions. C4 requires extra energy. The C4 pathway pumps carbon dioxide into bundle sheath cells but costs extra ATP, roughly 2 additional ATP per carbon dioxide fixed. When photorespiration is low, cool conditions, that energetic cost outweighs the small reduction in Rubisco oxygenation, so net carbon gain is lower than for C3 enzyme inefficiency. C4 enzymes and diffusion processes function best in warm, sunny environments and slow down in cold conditions. Result: C4 plants are less efficient than C3 plants in Uganda's highland areas. Effective temperatures on the productivity and abundance or distribution of C4 and C3 plants. C3 plants. In hot lowland regions such as Pakwach, Arua, Busia, and Iganga, high temperatures cause Rubisco to favor oxygen, leading to photorespiration. This wastes energy and reduces the rate of photosynthesis. Thus, crops like beans and wheat struggle to thrive under excessive heat. Result: C3 plants are less efficient in Uganda's hot lowland areas. C4 plants like maize, sorghum, sugarcane, and millet flourish in these hot conditions. Their Cran's anatomy and PEP carboxylase enzyme minimize photorespiration and sustain high photosynthetic rates. Result: C4 plants are highly efficient in the hot lowland districts of Uganda effect of dry conditions. C3 plants. In dry regions such as Karamoja, Nakasongola, and parts of Teso, water scarcity causes stomata to close to prevent water loss. This limits carbon dioxide intake, reducing photosynthesis and increasing photorespiration. Examples: beans, Irish potatoes, and cabbage experience poor growth under these dry conditions. Result: C3 plants are less efficient in arid and semi-arid areas of Uganda. C4 plants are better adapted to drought because they can maintain photosynthesis even with partially closed stomata. Their high water use efficiency allows them to survive and yield well in dry conditions. Examples: millet, maize, sorghum, and sugarcane. Result: C4 plants remain efficient in dry and water-limited regions like Karamoja and Nakasongola. As we complete this lesson, this table summarizes the different factors that affect the productivity of plants. I have made another video interpreting scenario items about photosynthesis. Do not forget to watch the video, and we will prepare for more lessons.